welcome. We're delighted that you're all here. The idea for this workshop came about, I don't know, maybe almost a year ago now, in the context of the Innovative Genomics Initiative here at UC Berkeley and UCSF. We wanted to communicate the work that's going on, not only, of course, here in the Bay Area, but elsewhere on CRISPR biology in particular, to help introduce all of you to this incredible technology and to discuss with you how all of you are using it in experiments that you might be doing. And we really hope that this will be very much of an exchange of information, and we invite you to ask a lot of questions and kind of get to know us over the course of the week. So it's a great pleasure for me to be here, and what Dana Carroll asked me to do this afternoon was to give you an introduction to the world of CRISPRs. And so what I want to do, I'm going to do really three things. I want to, first of all, give you some of the background on the system that we began studying and how it led to the technology that I think you're all familiar with for using the Cas9 enzyme as an RNA-guided technology for genome engineering. So I want to give you some of the background there. I'm going to give you kind of a high-level picture of how this is being used in a variety of systems right now, and certainly invite your questions if you have them. I want to tell you a little bit about a very practical sort of question, which is how do we deliver this technology into cells? How are we actually going to use it therapeutically? And I'm going to show you one way that my lab is involved in employing this and thinking about delivery. But I'd love to hear, again, I hope we can run this more as a conversation. And then towards the end of my time, I want to share with you some other aspects of CRISPR biology that I think are also interesting, that kind of show you where this system is going and the kinds of additional technologies that might eventually come from it. So, and again, I really don't want to lecture at you for 2 hours. I would really love to have this be a conversation. So if you have questions, please don't hesitate to interrupt me. And I think we'll take a little break part of the way through and give everybody a chance to have a 7th inning stretch and come back refreshed. This is just my very generic introductory slide, and I really want to just start off with a very general comment about the field of biology, and I would say molecular biology in particular, which I think we've all been really influenced over the last several decades by our understanding of the structure of DNA, and more recently the sequence of DNA inside of cells. And so it's been really fun and interesting for me. I was a graduate student back when Sidney Brenner and other people were asking the question, should we sequence the whole genome of cells? And should we generate the sequence of the human genome? And now we've had that sequence data available for almost 15 years. And you can see the impact that it's had on, I think, all of the kinds of experiments that we're doing and the way that we think about biology. So, but the comment that I like to make is that, having that sequence is like, you have a library, you have a set of books in the library, and you can go and look things up. The challenge has been that up until recently, we haven't had a technology that enables changes to be made to those volumes, right? Or to individual words or letters inside of those volumes. And so when you think about, the genomic information that we have, we actually now have the understanding. If you imagine, the entire human genome, we can actually understand that there might be a single base pair change in the sequence of the DNA that leads to disease or leads to a particular phenotype. And this is true, of course, not just in humans, but other kinds of organisms. And what if we had a technology that allowed us to go into that library, that sequence, and with the precision of a surgeon, be able to make a single base pair change in the DNA, much as we do what we're very, very accustomed to doing using our word processing programs, right? Where you could actually change a letter or a word that would change the whole meaning of a sentence or an entire chapter or volume in that information, that set of information. And I think that's really what the CRISPR system does, is it provides a very widely accessible technology for doing this. And as I'm going to tell you very briefly, it came about through not a targeted effort to discover such a technology, but really through a curiosity-driven set of experiments that we started with the goal of understanding how bacteria fight the flu. How do bacteria use small RNA molecules to fight off a viral infection? And so here at Berkeley, this system was actually, had already come to the attention of scientists who were working on metagenomic sequencing of bacterial communities. And that was really the research of Gillian Banfield. So she's a faculty member here at Berkeley whose work focuses on understanding the kinds of organisms that grow in different environments, the kinds of viruses that infect those microbes. And through her research, her sequencing research, she was uncovering a number of sequences in bacterial genomes that had a very curious property, which is they had a set of short repeated DNA elements that occurred over and over. And in between those repeats were sequences, typically 40 base pairs in length or so, that in 2005, 3 different groups had pointed out that in these CRISPR loci in bacterial genomes, these sequences in between the repeats very often correspond to sequences found in viruses that infect those cells. And so Jill Banfield's lab was one of the first labs to be sort of noticing these sequences. And when she saw these publications, they all occurred, they were not papers in journals that I was reading at the time at all, but she noticed these papers and she called me up because she wondered whether in fact there was an interesting function for these repetitive elements that somehow provided bacteria with not only a record of past viral infections through the integration of these little sequences, but also a mechanism for defending the genome against those viruses. And what sort of made it look as though this could in fact be some sort of a system that used enzymes was that together with these repetitive loci were very typically CRISPR-associated or Cas genes that co-occur in the genome with these repeats, looking like some kind of a pathway perhaps in bacteria. And of course, many bacteria have these. And so it was, at the time, just sort of an interesting possibility that these might, in fact, be some sort of mechanism for defending against viral infection. And to tell you why Jill called me in particular about this, I wanted to take a very brief step back, and this might be too basic for this audience, but just in case, just to make sure we're all on the same page, I just want to point out that this is the sort of what we call the central dogma in molecular biology that, we all learned this when we were in, taking AP high school biology or our first course in college, is that, the DNA in a cell encodes genetic information that's employed in the form of, first gets, converted into RNA through transcription and then is translated into proteins. And of course, not all of the information in the genome of a cell is converted into protein, only some, right? And then the rest of it is used for regulation, or some of it's used to encode RNA molecules that are not actually used to encode protein. And that's really one of the questions that my lab has always been interested in over the last, several decades that I've been doing science now, is that, we've really been interested in the fact that in many cases, DNA encodes RNA molecules that don't get translated into proteins, but are actually functional in the cell at the level of RNA. And so one of the hypotheses that was emerging early on in the CRISPR field was that maybe these repetitive elements that form the CRISPR loci are actually transcribed, and those transcripts are employed in the cell as RNAs to help protect the cell. And this was sort of an idea that came about partly because of familiarity with the RNA interference pathway in eukaryotes that our lab has been interested in for a while. And so what happened next was that over the next several years, there were just a handful of labs around the world, including us, but several other groups that did key experiments, both genetically and biochemically, to show that in fact, these CRISPR loci are bacterial immune systems. They're adaptive immune systems in bacteria. And this is kind of an overview of how these systems are thought to work. And so the idea is that in cells that have a CRISPR locus, like the one shown here, these systems enable cells to detect foreign DNA, and I'm showing it to you here in the form of a viral infection, but it could also be a plasmid transformation or some other detection of foreign DNA molecules that somehow get access to the cell. And these molecules are then somehow chopped up into bits that some of which are then integrated into the CRISPR locus. And as I'll show you a little bit later, this integration event is also a very carefully regulated pathway that involves some of the CRISPR-associated enzymes. And then these loci are transcribed into RNA molecules that are ultimately processed such that each of the little processed bits of RNA from the CRISPR sequence includes one of the elements that is coming, that has been integrated from a virus. And that RNA then assembles with one or more proteins that are encoded by the Cas genes to form RNA protein targeting complexes. And these use the information in the RNA to base pair with matching sequences in DNA. So you can see that if you have these RNA protein complexes pre-assembled in the cell, they're actually functioning as surveillance complexes. They go around the cell and they're basically sampling DNA and looking for sequences that might bear complementarity to the guide RNA. And if they find a matching DNA, then there's a very tight association that occurs between this complex and the target DNA that enables one or more of the proteins in the complex to cut the DNA and initiate a degradation pathway for the DNA. So it's a really sort of a beautiful, elegant way that cells can acquire sequences from their invaders and then use that sequence information to protect the genome from those same invaders. And I want to just point out a couple things. So the very first genetic experiment that was done to show that this is actually going on was actually done at a yogurt company. So back in those days, Dannon yogurt was made by a company called Danisco, and two scientists there, Rodolphe Marengo and Philippe Horvath, were working together to understand. They're trying to figure out very practical, project. They were trying to figure out how to protect bacteria that are used to culture yogurt from phage infection, a big problem in the food industry. And they had come across these, at the time, kind of obscure papers about CRISPR biology, and they thought, well, if that's a natural immune system, then let's see if we can employ it to protect these cultures. And so they actually did the first genetic experiments in which they actually just disrupted a couple of the CRISPR-associated genes in the bacterium they were working on and showed that, in fact, when they did that, those cells became much more susceptible to phage infection, right? And so that was, I sort of love the fact that, it was a very practical project that led them to an insight that was clearly really fundamental in this field for sort of establishing that this is, in fact, an acquired immune system in bacteria. And then it was the lab of John van der Oost in the Netherlands, a microbiology lab, that did the initial experiments showing that you could assemble an RNA protein complex from components of the CRISPR system in E. coli, and that complex was involved somehow in targeting DNA. They didn't know how at the time, but it was clear that it could bind to DNA that had sequences that matching the RNA. So on our side, here at Berkeley, so after talking to Jill, we started a sort of a project in the lab that was initially focused on kind of this part of the pathway, understanding how these transcripts are made, how they're processed, how they assemble into RNA protein complexes, and how they interact with DNA. And that eventually led to the collaboration that Dana Carroll mentioned, which involved the group of Emmanuelle Charpentier, she's pictured right here, and two people one from my lab, Martin Janek, and one from Emmanuelle's lab, Christoph Chylinski. Later, also Inez Fanfara contributed from Emmanuelle's lab. But it was really these two guys initially here that teamed up to do a series of biochemical experiments to investigate the function of one particular CRISPR-associated protein called Cas9 that was a gene that at the time had been genetically implicated in the CRISPR pathway of an organism that Emmanuelle's group was studying called Streptococcus pyogenes. So this is a bug that is a human pathogen. It has a CRISPR system that has a single gene that called Cas9 that was critical for protecting those bugs from phage infection. And the question was, what's the function of the protein encoded by Cas9? And this really line of research then really led us to, started off as very much a basic science project trying to understand the function of what we thought would likely be a very interesting protein, we thought likely to be some kind of an RNA-guided protein. And this led to what I like to call the aha moment, the point in time for us when this project went from being a strictly sort of basic science research project to recognizing that we had information about an enzyme that could probably be used to harness that enzyme as an important technology. And this was the data that we got that showed that the function of Cas9 is really to recognize double-stranded DNA and cut DNA in a programmable fashion, a way that it can be programmed with small bits of RNA so that you can direct it to sequences of your choice, really. So I'm going to show you, I want to describe a bit how this works. And tomorrow morning, I'm going to talk more about the real sort of structural biology of this and mechanism of how we think this works. But there's a few aspects of that I want to tell you about today. And I want to first just show you this is actually a photograph of a 3D printed model of the Cas9 protein to show you what it looks like and how it operates as a programmable protein. So this is first of all, I want to point out that this is a 3D printed model based on an actual crystallographic structure of this protein RNA DNA complex. And it's a crystal structure that was solved by Martin Ginnek in his lab now at the University of Zurich. And Jacob Korn, who's the director of the Innovative Genomics Initiative here, was able to take that set of coordinates and modify it a little bit. So what he did was to extend the DNA strands that are in the actual crystal structure to represent what we think this would look like if this protein was actually sitting on a much larger piece of DNA. So you have to understand that we're imagining a little bit the trajectory of the blue DNA helices that are projecting out of this complex. Okay, but that being said, the rest of this in the center is all based on actual crystallographic data. So what are we looking at here? Well, the protein is the molecule in white. So this is a protein that has sort of almost like a clamshell shape to it. So it's got a central channel that binds to an RNA molecule, which is the orange molecule here. And this RNA would normally come from the CRISPR locus in a bacterium. And then it's got two molecular blades, two active sites that each cut one strand of the bound DNA molecule. So one of those active sites is over here, and one is on the other side. So it makes ends up making a blunt cut in the DNA. And on the next slide, I'm going to actually show you this with one of the great things about this 3D model is that we had it printed so that it has magnets that hold together various parts of the structure. So you can actually pull it apart. So this is showing it to you now with one of the active sites pulled out of the structure, so you can really see the central channel. And what I want to point out is that if you can really see here that the way this works is it uses the RNA to form an RNA-DNA hybrid. This is actually an RNA-DNA helical structure that runs right through the center of the protein. It actually forms a 20 base pair interaction with DNA. And we'll come back to this, but importantly, this involves actually opening up of the DNA double helix and insertion of the guide RNA to form this RNA-DNA hybrid. So there's that very critical interaction. And when that happens, so the DNA kind of opens up, and you can sort of see this here in this model, how the two strands of the DNA come apart right in that region. And somehow that actually positions the two active sites of the enzyme so they can make a double-stranded break in the DNA. So there's two scalpels, two blades in the protein that each cut one strand of the DNA, making a clean cut. So let me show you that now a bit more schematically, and I want to tell you a little bit more of the background to this and how it works in bacteria, and then how we were able to modify it to be a really nice technology for genome engineering. So this is a cartoon that represents some of the information that Martin Jinek and Krzysztof Chylinski initially got for how this protein functions. So in this picture, the Cas9 protein is shown in blue, and I'm showing it to you bound to a double-stranded DNA substrate. And you can see here that the DNA substrate is unwound right in the middle, right in the region where this piece of RNA that has the 20 nucleotide guide sequence is located. So here in this cartoon, what you can see is that there's a RNA-DNA hybrid that's formed on the protein, leading to unwinding of the DNA in that region. Here are the two very schematic, obviously, active sites of the protein that are poised to make a cut in the DNA. And something that we found early on, this was one of those things, you're doing biochemistry, so Krzysztof and Martin were working with purified Cas9 protein, and we were testing activities in vitro, and initially nothing was working. We were using RNA molecules derived from the CRISPR locus, like this one here, and adding complementary DNA molecules to the reaction in vitro, and it was dead. There was nothing going on. So it's one of those, scratch your head, negative result. Is my protein bad, or maybe this isn't what this protein really does, or what's going on. And then we actually made a really interesting observation, which was that Emmanuel's lab had discovered previously that this RNA, a second RNA molecule called trace-rna, is actually encoded in the genome of this bacterium adjacent to the CRISPR locus. And it has a sequence that is complementary to the repeat element in the CRISPR locus, such that when those sequences are transcribed into RNA, this part of the tracer molecule can base pair with part of the CRISPR RNA sequence here to form a double-stranded RNA interaction. And Martin and Krzysztof figured out that if you included the tracer RNA in this biochemical reconstituted reaction, they could then get very efficient binding and cutting of DNA. And so that led eventually to figuring out that, in fact, there's a critical interaction here between these two RNA molecules that leads to binding and recruitment of the Cas9 protein. And if we leave out the tracer RNA, the system doesn't work. So in nature, it's a dual RNA-guided protein, requiring both of these separate transcripts to be made. And what people have done subsequently is to look in lots of different kinds of bacteria, and it turns out that bacteria that have this type of CRISPR system always have this second RNA. They have this tracer RNA molecule. The sequence here can vary, but it's always complementary to this end of the CRISPR transcripts in the whatever bug it's coming from, such that you can form this kind of structure to recruit Cas9. So that was a critical thing to figure out. And then furthermore, Martin Jinek in the lab did sort of what biochemists do, which is that they start trimming away at things to try to figure out what's the minimal composition of this complex that I need to get DNA binding and cutting activity. And in the course of doing that, he realized that he could really trim away extra sequences that were on the ends of the CRISPR RNA transcript, this molecule here, as well as on the tracer, and led us to wonder whether we could actually simplify the system relative to what nature has done by creating a single guide version of these two RNAs. And so the idea was, could we simply link together these two transcripts by putting in a short sequence of RNA that would link together the 3' end of this CRISPR transcript and the 5' end of the tracer RNA to create a chimeric RNA molecule that would have both the guiding sequence targeting information on one end and the Cas9 binding information on the other end. And again, this was sort of part of this kind of proverbial aha moment of realizing that if one could do that, then we would have a two-component system, a single RNA and a single protein that could be put together and programmed to recognize a DNA sequence and generate a double-stranded break in the DNA. And so I just wanted to show you a couple of experiments that Martin did, biochemical experiments to test this idea. So one experiment, very sort of super simple experiment, but it was one of those moments in my career when I sort of had, hairs standing up on the back of my neck when I saw the result, was to simply take a double-stranded circular DNA, so a plasmid, piece of plasmid DNA, and Martin designed 5 different single guide RNAs that would recognize the sequences indicated by the red lines. And something I didn't mention earlier, but another aspect of the cleavage reaction that Martin and Christoph had come to appreciate was that in addition to the 20 base pair interaction with the guide RNA, the DNA target sequence also had to have a very short motif called the PAM sequence adjacent to the target site. So for this particular Cas9 enzyme, it's a GG dinucleotide. And so Martin selected sites in this plasmid that would simply be adjacent to a GG dinucleotide, and we tried 3 on this strand and 2 on this strand. And then to do the experiment, we incubated Cas9 protein programmed with one of these different single guide RNAs together with a second restriction enzyme, SAL1, that cuts about 60 base pairs upstream of this part of the plasmid. So you're doing a double digest of the DNA. And then just looking at the products on an agarose gel system, and we saw here that each of the little fragments of DNA released in this double digest corresponded in size to the position of cutting that was dictated by the single guide RNA in each case. So that was, really a moment when we realized we had a programmable protein, a 2-component system that could direct Cas9 to cut sites in DNA that were dictated by this recognition mechanism. Now, the other thing I wanted to show you was just another sort of biochemical experiment that Martin did at the time. So, we were sort of looking, this was before any crystal structures were known for Cas9, so we were looking at, sort of a cartoon-like representation of the protein that, you could already, from looking at the sequence, you could predict where the active site residues were likely to be based on homologies to other well-known DNA and RNA cutting proteins or domains. So one of these domains is called RubC, and in this, what's interesting in the Cas9 protein is that there are 3 domains that are essential to form the RubC cleavage motif, and they're actually found in 3 separate parts of the Cas9 protein. So they're not together in the primary sequence of the protein. They're actually quite far apart. And this is this protein is a fairly big protein. It's about 160 kilodaltons or so, well over 1,000 residues in the polypeptide sequence. And then the other cleavage domain is called the HNH domain over here. So we could see these by sequence homologies to well-known DNA and RNA cleavage proteins, but we didn't really know, of course, how they would be arranged with respect to each other in the 3D structure at the time. But from looking at these conserved motifs, Martin picked out individual amino acids, one in the RubC motif and the other in the HNH domain, that were likely to be in the active sites of these domains. And so he made mutant versions of the Cas9 protein that each contained a mutation at one or the other of these positions. So just changing these amino acids to an alanine in each case, hoping that we could knock out catalytic activity of one or the other of the domains and then ask, how does that affect the DNA cleavage reaction? And this is just, again, this is a sort of a biochemical experiment testing these mutants. And so what we did was to generate a 50 base pair DNA, double-stranded DNA target sequence, so just 2 oligonucleotides that were annealed together that had a sequence recognized by the guide RNA that we were using for Cas9 in these experiments. And then we could radio label either the strand that is complementary to the guide RNA or what we call the non-complementary strand, so the other side of the DNA duplex that isn't directly complementary to the guide RNA, and then test these different versions of Cas9 for cutting those substrates. And so what you can see here is that when we do this using the wild-type enzyme, we get cleavage of the complementary strand, we get cleavage of the the non-complementary strand. And then what we found was that if we made a mutation in, a point mutation in the RUV-C motif, we still got cleavage of the complementary strand of the DNA, but we no longer got cutting of the non-complementary strand. And the opposite was true when we made a mutation in the HNH domain. Now we saw no cutting of the complementary strand and normal cutting, or almost very similar cutting, of the non-complementary strand. And so that really suggested that this is an enzyme that really has two separable active sites, one of which cuts each strand of the DNA. And that becomes important later in the technology development because it's been possible then to make nickase versions of Cas9 that will cut one strand or the other of a target DNA. And in fact, because we know which active site cuts which strand, you can control which strand you'd like to have cut by using the appropriate nickase version of Cas9. But this is really the underlying biochemical experiment that gave sort of that mechanistic information.
Jennifer,
yeah.
Yeah. So does the fuzziness of the non-complementary strand, does that indicate that the cutting is not precisely at one base pair? Is it able to move one base pair to the left or right?
So, when we've subsequently gone, let me just point that out. So you can see that when we cut, when the HNH domain is doing the cutting, you get a very clean cut, and we always see that. So, very precise cut in the DNA. When the RUV-C domain is doing the cutting, then you see this sort of more ragged pattern. And when we've gone back and done sort of careful kinetic experiments with these these mutant proteins, what we find is that it looks like the RUV-C actually also generates initially a very accurate cut, and it is a blunt cut in the DNA, but over time you get this sort of trimming that goes on. And so it looks like there's a little bit of, it might be, we're not entirely sure if that's an activity that is part of the RUV-C domain itself, or is it actually another nuclease that gets in to the system and does some of the trimming. But that's very consistently what we see. So we think that initially there's a blunt cut that's made by both active sites cutting, and then there's, on this strand, there is some additional trimming that goes on, just a little bit, a few nucleotides. Yeah. Okay, so I wanted to just sort of summarize what's required for Cas9 to generate a cut and bind and generate a cut in a DNA target sequence. And the other thing I didn't mention that I want to point out here is that Martin did a number of experiments to figure out whether there was something important about the supercoiled nature of DNA that would be involved somehow in this binding reaction or the cutting reaction. And it could be that there's some contribution of supercoiling to cutting by Cas9, but it certainly is not a critical aspect of the reaction. So in other words, we can take double-stranded DNA targets that look like this. So these are basically just two oligonucleotides annealed together. And as long as they have, that contains a 20 base pair target sequence adjacent to the PAM motif, then this functions very nicely as a substrate for Cas9 that bears a guide RNA with a complementary 20 nucleotide sequence at the end of the guide. So that, by the way, this is always at the 5' end of the guide RNA. And that leads to formation of, we think, this kind of a structure that you might see referred to in the literature as an R loop. So you'll see this, R loops are mentioned in the context of RNA polymerases, for example. They do the same thing, right? They basically open up DNA and they generate a transcript. Well, Cas9, we think, forms a very similar kind of structure where you have a local unwinding of the DNA double helix with an RNA interaction with one of the strands of the DNA to form this RNA-DNA hybrid. And when I showed you the 3D printed model of Cas9, that hybrid is really running through the center of the protein, and there's lots of interactions that hold that together. And then that leads to positioning of the two active sites of the protein to generate what we think initially begins as a clean double-stranded break, and then, as we just discussed, what we call the non-target strand, the top strand, gets trimmed a little bit by either the RUV-C domain itself or by some other nuclease activity that comes along with it. right? So this is just showing that, if you have RNA-DNA complementarity and you have the presence of the PAM motif, that seemed to be sufficient to give rise to this complex and the generation of a double-stranded DNA break. So that's really a lot of that's sort of summarizing a lot of the early sort of biochemistry that was done to figure out how this protein actually works. So, how do we get from that to a technology? And so, while we were doing all of this research, of course, there was a lot of work going on in other fields that was focused on taking advantage of natural repair mechanisms in cells that lead to changes in the genome of a cell. And so when we published this research in the summer of 2012, we actually proposed that we could tap into that kind of that sort of whole line of research and natural repair pathways in cells to use the system for what we called RNA programmable genome editing. And I know Dana has already given you a really nice kind of introduction to repair mechanisms in cells, and I'm going to show you just one quick slide here that sort of summarizes a lot of research that's gone on. But basically, it's been appreciated for probably several decades that plant and animal cells in particular can detect double-stranded breaks that occur in the genome. These could occur naturally during replication. They also come about due to various kinds of mutagens or radiation. And if they occur, then cells have to be able to repair these breaks. And they do so typically by either a mechanism called non-homologous end joining that leads to repair of the break with typically a small insertion or deletion at the site of the break. Or if there's a donor DNA molecule present in the cell that has sequence complementarity flanking the site of the break, then this can actually be recombined into the DNA to integrate new information at the site of the break. And so, when I was a graduate student in the 1980s, which now seems like ancient history, probably to a lot of you, but back in those days, there was already a lot, an appreciation of these mechanisms, even though, the details weren't well understood. But it was already known that if you could introduce double-stranded breaks into cells, that they would be repaired and that you could actually introduce changes in the DNA of a cell by doing this. And so people like Peter Dervin, back in those days, a chemist at Caltech, was already, generating various kinds of chemical methods for doing this. And I was at Mass General Hospital when the first experiments were done to map the location of the Huntington gene, for example. And Jim Gusella did that by basically, chromosome walking, where they took advantage of, site-specific cutting of the chromosome with oligos that were hooked up to bleomycin, a drug that will cut DNA. So there was already sort of a really nice kind of pathway laid for doing this. And then over the years, more and more sophisticated methods were developed for doing this. And Dana, of course, was instrumental in one of the first sort of ways of doing this using proteins, called using zinc finger nucleases. And sort of these and talons and homing endonucleases, and there's a few other classes of these, are all proteins that can be designed or engineered to have very specific DNA recognition properties. So they can bind to DNA, and then by coupling those to cleavage domains, you could generate these sort of artificial nucleases that would have very specific cutting activity on DNA. And so there was already, really nice literature showing that you could use these kinds of proteins to do this, to trigger double-stranded breaks leading to repair pathways in cells. And that if you did it by targeting a sequence of interest, you could often engineer a site-specific change in the genome of cells. So very exciting technology. But I think of these as sort of hardwired technologies in the sense that they could work extremely well, but they did require a fairly sophisticated protein engineering pipeline. So you had to be able to engineer these proteins and then vet them and test them for each site where you might want to introduce a change. And the reason we thought that the Cas9 protein would be an interesting alternative to these is that it's more like a software-driven technology, right? So where the RNA is the software, it provides the programming, and the protein doesn't have to change in the reaction. So very trivially, one can engineer a new piece of RNA and allow it to form a complex with Cas9 that would direct this kind of activity. And of course, you all know that really took off. And so within about 6 months of that initial publication, papers began to appear showing that you could actually use this technology to engineer changes in all sorts of different kinds of cells. Initially, this was done in mammalian cells, and then it was also shown that you could use this to engineer whole organisms like zebrafish. You could use it in all sorts of cells like stem cells. You could use it, of course, in bacteria, and then plants, and many, many other kinds of cells. And in addition, one of the sort of cool things about this technology is that it's great not only for introducing covalent changes to the genome, but also by taking advantage of mutant proteins like the ones that I described to you that allow nicking of DNA. You can also engineer this to do other things, such as control transcription. And so you'll hear later this week from Jonathan Weissman and people from his laboratory that did a lot of these initial experiments where they were able to actually engineer out the cutting activity entirely for Cas9 by making point mutations in both of the cutting domains. And then you have a very nice RNA-programmed DNA binding activity. And by coupling that to transcriptional activators or repressors, it was possible to use this to very precisely control transcription in cells. And you'll hear a lot more about that later in the week, how that's done and sort of the current state of that technology. And another sort of, I think, application of all of this that I would say is very much up and coming. There haven't been too many papers on this yet, but I'm aware of a number of groups that are actively working on this, is to use this platform for localizing and visualizing particular regions in a genome and do this in living cells. So, there are lots of Rob Singer and others have developed nice technologies for localizing and imaging regions in the genomes of fixed cells. But what if you could actually do that in a living cell? And this technology actually enables that. So I think that's an area where there's a lot of interest and, I think lots of interesting kind of technology development to try to turn that into a robust kind of platform for cellular imaging, live cell imaging. So I want to, first of all, let me ask how we're doing. Any burning questions at this point? Yeah, in the back, yeah.
I'm wondering, for the non-homologous end joining, are there different types of ligases, like for that, some of them have, some variants have the tendency to remove a base pair, some of them have, so you can actually exploit that, kind of either, add or remove, rather than just, see what happens?
There's, again, I would say Dana is probably the better person to answer those kinds of questions, but I'll tell you what I know, which is that, there's a lot of interest in this and lots of groups that are, I think one of the exciting things about this technology is that it now actually enables research on these repair pathways to be done in a very controlled way that, maybe more so than has been possible in the past. And so there are a number of groups, including here at Berkeley, that are involved in trying to really understand exactly what you're asking. In other words, what are the contributions of different kinds of repair enzymes in cells? Can we really eventually get a handle on the kind of repair that happens after a cut is made? I think this would, this is one of the really exciting forefronts of the technology right now, is if we can not only control where the cut is made, but also how is it repaired. This would obviously be really, really important. And so, that's, I don't know if you want to add anything to that, Dana, but, I think there's a lot of interest in this. And I feel like, from what I'm hearing from various groups that are involved in this, I think we're likely to see a lot of progress on that question in the next, few months to a year. So let me show you, there was another question here, yeah.
Does the PAM sequence need to be in the target sequence? That NGG has to be?
It has to be there.
Does that limit where you can make a cut and find that NGG?
That's correct, yeah. And I'll have more to say about that in a few minutes about how we think that actually works. I also think that it's going to be different for different Cas9 proteins. Nature has come up with a wide range of these things in different bacteria, and they seem to have different PAM specificities. We also think that based on recent data that we haven't published yet, that there will be PAM-independent DNA cutting activity that is more or less predominant in different Cas9 enzymes. For this one, it's very PAM dependent, but there may be other Cas9 proteins where that's not as much the case. I would like to show you I want to show you a little movie that just kind of pulls together a lot of the things that we've been kind of discussing so far and shows you a kind of an artist's rendition or imagination of what an editing reaction would actually look like in a cell. We're zooming into a cell, and we're going to go inside the nucleus. And you know that if we're looking in a eukaryotic cell, of course, the DNA is highly packaged, right? In chromatin, we've got individual nucleosomes that you can see here. We absolutely don't know how Cas9 deals with nucleosomes yet, but we know that somehow it does get access to the DNA. When it finds a site that has complementarity to the guide RNA, that allows this protein-RNA complex to form the structure that I showed you previously and to cut the DNA. So it makes this blunt cut or perhaps slightly staggered cut in the DNA that now has to be repaired. And this is, of course, not accurate in terms of the mechanism, but the point is that new information can be integrated at the site of the break. And of course, what's really exciting is that this reaction seems to work in all sorts of different cells: animal cells, plant cells, fungi. I've heard about it working in all kinds of parasites. It will work for cutting viral DNA. So it's really a very general kind of reaction. And one of the things that we'd love to understand better is how does the complex really get access to chromatin. And I'm going to show you some data that's unpublished so far from a collaboration here at Berkeley that shows some of the information that we're getting about how that actually works in a living cell. So let me show you one experiment. I want to just show you sort of an experiment that was done by Russell Vance here at Berkeley early on. So this was really when, I think, for me personally, the real power of the technology kind of came home to me. I was very excited about this, and I was thinking about various kinds of applications of genome engineering that one could do using this technology. And then I think this was maybe sometime early in 2013. I was walking across the Berkeley campus, and I was chatting with Russell Vance, who's one of our immunologists here on the faculty. And Russell said, I really think we should be testing your Cas9 system in mice. We're running this mouse lab, and wouldn't it be great if we could use this to make transgenic mice much more quickly than is the sort of traditional route, which takes a year or so. And so he sent a student over to my lab, and I gave them a few constructs, and then I didn't hear from Russell for a few weeks. And then he contacted me by email, and he said, I want to show you an experiment that we just did using Cas9 to engineer transgenic mice. And so the experiment they had done was to basically take fertilized mouse eggs. You see an example right here. So it's being held by a pipette. And coming in from the other side is a needle that is injecting, in this experiment, the Cas9 construct and appropriate guide RNA into this early mouse embryo. And so the experiment was to inject these fertilized eggs and then to implant these into female mice and allow them to develop into mice and see if the targeting that was intended through the design of the guide RNA actually worked for generating a transgenic mouse bearing that genetic change. And this was the picture that showed up in my inbox from Russell. And basically what he told me was that the way they had set up this experiment was to take a strain of mice that have normally have a black coat color, and they targeted a gene called tyrosinase that is essential for creation of this black pigment in the mice. And this was one of the litters of mice that was born doing that experiment with this injection that I showed you on the last slide, where 6 of these 8 mice were white. And furthermore, when these mice, the DNA was sequenced, they found that they had a gene disruption in the tyrosinase gene at exactly the place where Cas9 induced a cleavage event. And you can also see, I took the slide out, but I have another slide that actually shows that in another litter of mice born after doing this kind of experiment, a couple of the animals were chimeras. So they actually had black and white coat color, right? So, but in this experiment, these were all turned out to be homozygous changes in the gene. So, you know, this really was exciting to see, and it had only taken a few weeks to generate these mice, and they turned out to be perfectly normal mice, except for this genetic change. And when Russell's lab did sequencing of the sort of potential off targets in these mice, they didn't detect off-target events. So it looked like these mice were basically normal, except they had this genetic change. And so I showed this picture to members of my lab. And you know, my lab is a lab full of biochemists and people doing structural biology, people who don't think about animals directly very much. And half of the people in my lab said, wow, that's really cool. And the other half in my lab said, that's really freaky, right? Because I think we all recognize that, you know, it was in a way trivial to do this. I mean, this was done by graduate students who no doubt are very talented, but nonetheless, they're not, you know, they haven't had, you know, 25 years of experience. They were able to basically take constructs and within a few weeks get this kind of experiment to work. And if it works in a mouse germline, it probably would also work in the human germline, and, you know, that has a lot of implications, right? So, and then I just wanted to point out that, you know, now there are, I think, well over 1,000 research papers in the literature that have come out since early 2013 using this approach to engineer all sorts of cells and organisms. And I'm just going to show you a few examples here. And of course, over the course of the week, I'd love to hear from any of you that want to share it, you know, the kinds of things that you're interested in doing. But, you know, lots of work going on in plants. So Brian Staskowitz and various of his colleagues here in the Bay Area, plus, of course, many others, are using Cas9 to engineer plants for research and other kinds of applications. We can do this in fungi. Groups here in the Bay Area at the Energy Biosciences Institute, for example, have been engineering fungi to introduce new metabolic pathways that allow them to metabolize cellobiose and other substrates that have traditionally been very challenging to use for making biofuels and other kinds of chemicals. And then, of course, all sorts of animals. There was recently a paper talking about engineering pigs. There's lots of interest in this because one could, in principle, engineer pigs to have more human-like organs that would allow transplants to be done more easily. And of course, there's a company in the Bay Area that's also interested in engineering companion animals, so making cats that are hypoallergenic, for example, using Cas9. You laugh, but apparently it's a big market. I don't know. They raised a lot of venture money to do this. And then, you know, just many, many applications that are very directly relevant to human health. And, you know, this includes not only engineering stem cells for therapeutic applications, but also creating better animal models of human disease, both in rodents, but also potentially in monkeys. And there's a group, at least one group in China, that's already published experiments where they've been able to engineer monkeys, and there's clearly more to be done there. And then we think ultimately it'll be possible to use this directly in humans for therapeutic applications. And there already are many companies ranging from startups to big pharmas that are very excited about opportunities there. Okay, how am I doing here, Dana? We're sort of 50 minutes in. Yeah. How are you guys feeling? Shall we go a little bit longer and then maybe we'll take a break in about 10 or 15 minutes? Does that sound good? Okay. So what I'll do then is share with you some more sort of basic research that's going on in our lab to try to understand the answer to this question, which is how does Cas9 find target sites in genomic DNA? And we're interested in this not only because we're curious about it, but we also really think that by understanding the mechanism of this system, as I've already showed you, that proved to be very important for developing this as a technology. But we think that it's also going to be really important to understand how it really works if we want to make sure that it's accurate enough and efficient enough and ultimately safe enough to be used for the kinds of applications that we've been talking about, especially for human therapeutics. But again, for us, this always begins with molecules, you know, thinking about individual molecules, how do they work, how do they operate? And so early in 2012, we actually started a collaboration with Eric Green's lab at Columbia to begin to address this question. And we wanted to take advantage of a system that Eric's lab had developed for single molecule imaging of proteins that interact with DNA. And this is a system that he likes to call DNA curtains. So he actually had come to Berkeley and was showing data for experiments like this, where they were actually taking phage lambda DNA, so these are 48 kb molecules of DNA, and they can be attached to a slide on one end of the DNA, as shown in this experiment, and then visualized using a green dye. So you can actually see these individual strands or threads of the curtain are each an individual 48 kb molecule of DNA. And then to look at proteins and how they interact with these strands of DNA, we can take protein, in this case we're doing it with the Cas9 protein, and introduce a quantum dot label that allows visualization by a technique called TIRF microscopy, total internal reflection microscopy. And so in this experiment, what you're watching is DNA molecules that are, when we turn on the buffer flow, we have buffer flowing across the slide from top to bottom. These molecules are extended. When we turn off the buffer flow, these molecules snap back up to the point of attachment on the slide. And that allows us to see where on these DNA strands these individual Cas9 proteins are interacting. These are the magenta colored dots that you can see on the picture. And what you can notice is that when the DNAs are extended, many of those individual Cas9 proteins are lining up on the DNA at about the site where they should, based on the guide RNA that we're using in this experiment to recognize a particular sequence along the length of the lambda DNA genome. And so Sam Sternberg, a student in the lab doing these experiments along with Cy Redding in the Green Labs, two great students, great collaboration where Sam was providing the biochemical and sort of biological knowledge of the system. Cy Redding is a physicist who was an expert in setting up these kinds of experiments physically and interpreting the data. So it was a very, very good sort of partnership between those two guys. And they could show that they could actually program Cas9 to recognize sites along the length of the lambda genome and get sort of data that look like this. So it's sort of a control experiment here, just showing that in this kind of single molecule system, we could get very nice targeting of individual sites along the length of the lambda DNA. And you can also see that the resolution is, it is what it is, right? It's not at the level of a few base pairs, right? There's some spread to it, but nonetheless, we could really see very nice evidence of site-specific interaction with the DNA in an RNA-programmed fashion in these experiments. And so I just want to show you a couple of things that came out of this. So, of course, in addition to looking at these actual on-target binding events by Cas9, we also were paying attention to these off-target events. And what we found was that in contrast to on-target binding, and I'm not actually showing you the data for this here, but for on-target binding, we saw evidence that Cas9 binds to a place in the DNA and then never lets go, right? And this is all initially just using a catalytically inactivated form of the protein. So it looked like very long-lived binding to on-target sites in the DNA. And that's been quite consistent with what we've seen in other experiments that I'll share with you in a few minutes. But we also saw events like this, where you saw very transient binding to DNA at sites that did not correspond to where the guide should be interacting with the DNA. So very, very transient binding events. And by mapping where those occurred along the length of the lambda genome, which is plotted in this plot along the x-axis, so this is the position along the length of the lambda genome, and on the y-axis is the probability of a binding event at a particular position. And you can see that in this sort of a plot, we saw an overabundance of events in this half of the genome relative to this one over here. And that was sort of interesting because just fortuitously, it turns out that this half of the lambda genome is more GC rich than this one. And that made us think that that was sort of interesting in the light of the fact that the PAM motif for this protein is NGG. And if you plot this out, you get a plot that looks like this. It's not a great correlation, but it's also not random either. So it really looked like there might be an interaction with PAMs. And we started thinking that maybe what we were calling non-specific binding was actually specific, but it was specific for PAMs. And that might be driving the initial interaction of the protein with DNA. And so Sam Sternberg in the lab came back and did a bulk biochemical experiment to really test this. And he did this by using what we call a competition cleavage experiment. And the way this works is that we take a radiolabeled double-stranded DNA substrate, so it's radiolabeled at one or both ends. It's got a target sequence and a PAM motif in it. And when Cas9 is programmed with an appropriate guide RNA, of course, it can bind to this substrate and generate a cut, and I've showed you that already. And then to do the competition, we can include different kinds of DNA molecules that are not radiolabeled in the reaction that could potentially compete for binding to Cas9. And if they compete for binding, then they effectively block these radiolabeled substrates from interacting productively with the protein, and we would see a decrease in the amount of cutting of the radiolabeled substrate. And just to show you sort of a control for how this works, if we do this where we use the DNA substrate itself as the competitor in this reaction, so it should be a great competitor, right? Then what you can see in these reactions down here, you're looking at radiolabeled DNAs, and these are little time courses, 5-minute time courses of cutting by Cas9, and using different concentrations of the unlabeled competitor, which in this case is just this substrate DNA in the reaction. And the more you add of the competitor, the less cutting you get of the radiolabeled molecules, right? And so we can actually get a sort of a rate constant for that kind of competition in this sort of experiment. So, but here's the actual experiment. So the actual experiment was then to say, all right, what if we use a substrate and then competitors in this reaction that have different numbers of PAM sequences. And the idea was if PAM interaction is really driving Cas9 binding, then we predicted that it should be the case that even if you don't have any target complementarity at all in the competitor DNA, so there's no place for the guide RNA to interact with this competitor DNA. But if we had potentially an increasing number of PAMs, these should in principle be increasingly good at competing for binding to Cas9 and competing for cutting of this radiolabeled substrate. And that turns out to be exactly what we saw. So this is Sam Sternberg's data. So what he found was that, depending on this is the concentration of the competitor down plotted here as a function of this K-cleave value. And what you can see is that the larger the number of PAMs in the competitor, the better a competitor in the reaction it really was. So this number 4 here is this one, right? So it actually really blocks, cutting of the radiolabeled substrate because it's actually a really good competitor for binding to Cas9 compared to the substrate or these DNAs that have fewer numbers of PAMs, even though there's no target sequence in any of the competitors. And then I think the most telling competitor was this one here. So then Sam said, well, if that's really true, then I should be able to use, as a good competitor should still, could still contain the target sequence. So that's shown in this competitor right here. So it's got the full 20 base pair target sequence. It just has a point mutation in the PAM motif. And that DNA turns out to be as good of a competitor in this reaction as this molecule that had lots of PAMs in it, right? So it really, and so it really suggests that the way this protein interacts with the substrate is initially by binding to the PAM motif. And we have lots of other evidence now that that's really the case, also based on crystal structures, and I'll show you some of that tomorrow. And I just want to summarize. So these are really I would say, the key conclusions that came out of that work. First of all, that binding occurs first at these PAMs. But we also found that there's very high affinity binding not only to the substrate in the reaction, the DNA, but also to the product. So there's no turnover of substrate. And I think that's an interesting point that gets back to thinking about how repair happens after the cut, right? If this is really happening in cells, and we have some evidence that it is, then you could have a very high affinity complex that includes Cas9 binding to the site of the break in the DNA, and it might require some kind of active removal before repair could really occur. And finally, just that we think that PAM binding also triggers Cas9 catalytic activity. There's evidence that, that there's actually a structural rearrangement in the protein that triggers cutting only when this is associated with a fully sort of complementary target sequence. Okay, and so this is just showing kind of the model that we have currently for how this works. So the idea, and this is all, again, right now based on in vitro data that I've showed you, but I'm about to show you some more sort of in vivo stuff, but this is based on in vitro data. The idea was that Cas9 RNA complexes are binding and releasing DNA, perhaps with very fast on and off kinetics, and that upon interacting with a PAM sequence, this might slow down the rate of release of the DNA such that, basically to provide time for this complex to interact with the adjacent sequence in the DNA, and really suggested that the first few base pairs adjacent to the PAM in the DNA might be a critical interaction to make with the guide RNA. So it might be critical to have complementarity there. And if there is complementarity, then that allows formation ultimately of the kind of R-loop structure that I showed you before and positioning of these active sites to generate the double-stranded break in the DNA. Okay, so maybe what we'll do now, well, first of all, let me ask if you guys have questions at this point. Okay, and if not, maybe let's just take a few-minute break and stretch your legs, and then let's come back in about 5 minutes and we'll carry on. I want to now sort of, starting with sort of this model that I showed you that's based on in vitro data, I want to show you a few slides that represent unpublished data that we have for the behavior of Cas9 in live cells. And this is something that has been really initiated through a collaboration between my lab and the lab of Robert Tejan here, as well as his colleagues at Genelio Farm Research Campus in Virginia, to use really state-of-the-art cellular imaging methods to investigate how Cas9 behaves when it's in the nucleus of a living cell. And so this is work that was done by Spencer Knight, a graduate student here at Berkeley. And what Spencer did was, he looked at all the in vitro data that we had, and he said, it'd be great if we could actually understand, take the model based on in vitro data and really test it in living cells. Is that really representative of the behavior of these particles when they are searching through the genome of a real cell? And so to do that, what he did was to generate constructs that look like this, where we have a deactivated or D form of Cas9. So this is catalytically inactive Cas9 that can still bind to RNA and recognize DNA in that same sort of programmable fashion. And he's got this protein fused to a nuclear localization signal, as well as a small protein domain on the end called a halo tag that is the structure of this domain is shown over here. This was actually published in a paper in 2008 in ACS Chemical Biology, where a group had been able to take this domain, which is normally as a dehalogenase, and generate a mutant form of this domain that gets trapped in the middle of its reaction. So it basically leads to a covalent ligation of substrates that look like this to this domain. And so if you have this domain fused to your protein of interest, you can actually introduce this into cells, and then along with the fluorophore here, and in cells, this will interact specifically with the halo tag to form a fluorescently labeled version of this fusion protein. And you can do this in living cells. And this has actually been done with a number of other kinds of cells, a number of other kinds of proteins. And what Spencer did was to generate a mouse cell line that is stably expressing this fusion. We can then just put in these dyes and generate fluorescently labeled Cas9. And then we can actually follow its behavior in these cells. So the data that I'm going to show you is using this halo tag system to visualize Cas9. So The first thing I want to show you is just sort of what some of these data actually look like. So here's the cell, and we're actually following the behavior of these particles inside the nucleus. And as I mentioned, so we have the Cas9 halo tag fusion being stably expressed in the cells. We also express stably in these cells a protein called HP1, which is a heterochromatin protein 1 fused to GFP, so we can actually label a protein that is associated with heterochromatin, and I'll show you why that's useful in a few minutes. And then we transiently express guide RNAs in such a way that we can easily change the sequence of the guide RNA to reprogram Cas9 and then watch its behavior. And so this is what, just some of the initial raw data look like, where you can actually see these particles of Cas9 moving around the nucleus, and we can take snapshots at different frequencies, ranging from a few milliseconds to longer, to try to watch different types of behavior that these particles exhibit. And so, so one of the things that we noticed was that the search mechanism of these complexes in cells really seems to be dominated by what we call 3D diffusion. And so what I mean by that is that, and I didn't mention this earlier for the in vitro data, but all of our in vitro data with the DNA curtains and sort of watching the behavior of Cas9 and those sorts of single molecule systems suggested that this is not a protein that gets onto DNA at one end and then slides to a target, right? We had no evidence that it really does that. And so we had sort of, and that's why I proposed to you this model where we have very rapid binding and release of DNA by Cas9. And we think the same is really true, sort of is consistent with the behavior that we see in living cells. And so in these experiments, what we're doing is, in this case, we're using 30 millisecond exposures of the cell. We're looking at different positions in the nucleus and then doing a reconstruction so that we get sort of a three-dimensional view of where these particles are going as they move around the nucleus. And in this experiment, we actually programmed Cas9 with what Spencer calls a nonsense guide. So this is a guide RNA that shouldn't recognize any particular site in the mouse DNA genome. And so when we do that, we get, we sort of can see the general behavior of these particles when they're searching through the cell, but they're not actually localizing at any particular place based on sequence-guided targeting. And so what does that really look like? And so what we did was to initially just to plot the behavior of these particles that are, this is actually using just the Cas9 protein alone, no guide RNA added to the cells, to see how this protein alone interacts with DNA. And we're plotting the log of the diffusion coefficient here on the x-axis and the frequency of particles with this particular kinetic behavior on the y-axis. And we get a plot that looks like this for Cas9 protein alone in these cells. And just for comparison, this is sort of what you would see for molecules that are freely diffusing through this medium. And this is where we would see the sort of the diffusion coefficient for particles that are interacting with in nucleosomes. This is a histone protein, H2B. And so in general, what we found was that these particles are moving very quickly around the nucleus relative to other DNA binding proteins, and especially for histone proteins. And so then, of course, the question is, well, how does that change when you reprogram Cas9 with a guide that is recognizing a site in the genome? And the challenge here, and this is just sort of a limitation of the current technology, is that we'd love to watch behavior as Cas9 goes to a particular single site in the genome, but we don't have the resolution to do that right now, right? And so the strategy that we came up with was, well, let's actually program Cas9 with a guide that will direct it to many sites, and then we can basically watch a change in the bulk behavior of a lot of these particles as they are directed to many sites in the genome. And so we picked a site that called a sign element, sign B2 in particular, that is present in about 300,000 copies in the mouse genome. And so the idea here is that, again, we program this with a guide that should allow it to interact with many sites, and then we can watch whether there's a bulk change in the behavior of these particles. And so when we do that, Okay, so first of all, this is what happens when you use this nonsense guide. So there's no particular site that this guide should be directing Cas9 to. And it looks like the behavior of these particles is very similar to what we see for the protein alone. But if we program Cas9 with this B2 sign recognizing element, RNA, now you can see that many of these particles have a much slower rate of diffusion around the cell. And in fact, it almost looks here like we have biphasic behavior, right? We have some particles that are still have this very rapid kinetic behavior, but we have a lot of the particles that are now moving much more slowly around the cell. And so this kind of experiment really allows us to start to ask questions like, how long does it take one of these particles to find a particular target sequence in the genome? And also, how long does it spend when it gets there? Okay, right. Okay, so let me first show you this. So one of the experiments that Spencer did once he had this system kind of working was to modify the sequence of the guide part of these RNAs such that we introduced mutations between the guide sequence here, this is the RNA, and the target site in the DNA, either 13 or more base pairs into the guide sequence, and this is measuring from where the PAM would be in the DNA target, which would be over on this end, or immediately adjacent to where the PAM interaction occurs in the DNA, and then ask how those guides affect the behavior of Cas9. And you can just see here by comparing these plots, this is the one that I showed you before with the nonsense guide, and then here's the one on the bottom with the fully complementary guide recognizing this B2 sign element. If we use a guide that has mismatches to the target sequence immediately adjacent to where the PAM interaction occurs in the DNA, that actually, those particles behave very similarly to the nonsense guide. In other words, it looks like it's very important that these initial base pairing interactions that occur between the DNA target sequence and the guide RNA are actually complementary, right? So even if we have full complementarity of the rest of this sequence with the target, if those first few nucleotides are mismatched to the target, then it really looks like those particles behave as though they're not interacting with the target at all. And if we introduce mismatches within the sort of in the middle of the target or closer to the end of the guide sequence opposite where the PAM binding interaction occurs in the DNA, then you can see that actually now there is at least some behavior of these particles consistent with target interaction, although we don't know for sure, whether those, because we're not looking at individual events, we can't be sure whether those are, really binding to those targets, or is it just representative of a slower release of these sort of close to matching target sites. Something that we'd love to get at with, we'd love to sort of have guides that allow us to interact with fewer numbers of targets eventually, so that we can really start to get at more quantitative aspects of that question. I want to show you this experiment here. So this is an experiment called FRAP, or fluorescence recovery after photobleaching. And this is an experiment that you can do where you basically are looking initially at the nucleus of a cell, and we have some fluorescently labeled Cas9 proteins in there, many of them. And we can actually shine a laser in one part of this nucleus such that we photobleach those particles. And so they get, and I'll run this as a movie. You'll see this in a moment, where you bleach out a particular part of the nucleus, and then you can just watch how long does it take for other fluorescently labeled particles to encroach on that region of the nucleus, and sort of you get recovery of the fluorescent background or signal after photobleaching that spot. And I think this, yeah. So here we are sort of running this experiment, and I think the bottom will show this, right? So we can photobleach initially, you see a sort of a blot there, sort of a dark spot, and then over time, you can see that we start to see particles that are fluorescently labeled that encroach on that region. You can sort of plot the kinetics of that behavior, and it helps you to understand how fast, what really are the kinetics of the behavior of these particular particles. And we can do this either with Cas9 programmed with a nonsense guide or with Cas9 that's been programmed with this B2 sign recognizing guide element. And basically what these experiments told us was that Cas9 has, we think, a fairly long lifetime on target DNA and a pretty short lifetime on off-target sites in the DNA, such that we could sort of calculate roughly that when Cas9 interacts with non-target sites in DNA, we think the lifetime is less than a second versus many minutes when it's interacting on an actual target sequence. And of course, there's probably some variation. If you use guides that are close matches but not perfect matches to targets, then there could be some intermediate values. And that's something that in the future we'd really like to get at that more quantitatively. But I think very consistent with what we see in vitro for interaction with DNA. Okay, and then I just want to show you this one other experiment. So one of the things that comes up a lot, and depending on the kinds of applications that you guys are thinking about, this could be relevant to your experiment, is how does Cas9 deal with different kinds of chromatin? So of course, there are more and less compact regions of the genome, depending on whether they're in heterochromatic regions or euchromatic regions. And a lot of data now is available for targeting different sites in genomes, and it really has emerged that there doesn't seem to be a strict cutoff. In other words, people have done experiments where they're targeting genes that are found or sequences that are found in heterochromatin, and they still seem to be accessible by Cas9. You can still get editing in those regions, although perhaps with lower efficiencies than in euchromatic regions. So we wanted to do the following experiment to try to actually look at the ability of Cas9 to get at different parts of the genome. And so the way this was done was to take advantage of this HP1 GFP fusion protein that's being stably expressed in these cells. So this is a protein that's associated with heterochromatic regions, and because it's fluorescent in these cells, then we actually have ways to label parts of the genome that are heterochromatic. And then we can program Cas9 with guides and watch how it interacts with heterochromatic regions of the genome versus parts that are in euchromatic regions. And okay, so this is just showing what these cells look like. So if we have the stable expression of HP1 fused to GFP, we can see these sort of bright regions, blobs that correspond to heterochromatin in the cell. And I'm going to show you, this is sort of what raw data look like when we do single particle tracking of Cas9 guide RNA complexes as they move around this HP1 GFP expressing nucleus. Doesn't look like much. It's hard to really tell what's going on. But what's really cool is when you take snapshots of these particles as they move around the nucleus over time and then trace where these tracks are going with respect to heterochromatin, what you see is a plot that looks like this, where we see many tracks for these particles that are outside of the heterochromatic regions, but we do see a fair number of forays into the heterochromatic regions of the genome. And that seems to be very consistent with what we've observed for when we introduce Cas9 guide RNA assembled protein RNA complexes directly into cells and watch the sort of, we can measure the time it takes from first nucleaffecting those into cells to when we first observe evidence for editing, and it's just 4 hours, right? So it's really very fast. If we target those particles to heterochromatic regions in the genome, it's more like 8 hours, but we still see editing occurring, right? So it just looks like it happens, but it's slower. And I think that's consistent with what we're seeing here, where you still see some ability of Cas9 somehow to get into these very compacted regions of the genome. And of course, going forward, we really would love to understand better how that really works. Okay, so let me say, I want to now just say a little bit about delivery of Cas9, and this really gets to sort of this question of, if we're going to use this technology ultimately in therapeutic applications, we have to be able to get it into cells or tissues where it's needed. And ultimately, I think, it would be fantastic if one could really envision doing this in a way that allows us to treat adult patients where we can deliver this editing technology just to the cells or tissues where we need to edit the DNA or make some kind of modification to the genome and not have to expose other tissues to this. So how does one do that? And as you guys are probably all aware, there's different strategies for doing this. One can introduce Cas9 and guide RNAs encoded in DNA. You can do this in the context of a virus, for example. So you could imagine having vectors that give you tissue-specific delivery. And one of the challenges there is the size of the Cas9 protein. And of course, there are smaller natural variants of Cas9 that people are working with that might help get around that issue. But nonetheless, you still have to, this still requires assembly of the complex in the cell, and how efficiently would the guide RNA survive so that it can assemble with Cas9 is a question. You can also deliver these components in the form of RNA, so the guide RNA can be introduced into the cell as a pre-made transcript along with messenger RNA that encodes the Cas9 protein. Again, you have to, this requires assembly of the complex inside of the cell. These components have to find each other and they have to assemble efficiently. Or you could do this. You could actually just pre-assemble an RNA protein complex as an RNP or a ribonucleoprotein and deliver that to the cell. And if you could do that ultimately in a tissue-specific fashion, then you would have your editing tool already pre-assembled going into tissues, doing the editing. And one of the things that's sort of appealing about this idea is that this would have a certain lifetime in cells that would be a function of whatever the stability is of the protein and the RNA, of course. And then it would be degraded. And so potentially, if editing were relatively fast, you could imagine that you get this into cells, editing occurs, and then this complex is degraded and doesn't stick around to cause problems with off-targets and perhaps other toxicity issues and things like that. So we were sort of attracted to this idea also because we're biochemists, right? So we're very comfortable working with these kinds of samples, and we thought, wouldn't it be cool if you could actually just introduce those directly into cells and see evidence of editing? And I just want to show you a couple of slides about this. So this first experiment, and this is all done by initially by two postdocs in my lab, Steve Lin and Brett Stahl, who've teamed up to do a lot of this. Another sort of great collaboration because Steve is a biochemist by training and works, very comfortable working with purified proteins. Brett is a terrific cell biologist who's comfortable growing up all sorts of cells. So the two of them teamed up to test these ideas initially. And basically what they found, so what are we looking at here? Well, we're basically looking at sort of a standard type of assay that people use to test editing efficiency at particular sequences in genomic DNA that's based on PCR. And so this is called the surveyor assay or the T7E1 assay. And you can basically amplify a particular locus in DNA. In this case, we're looking at the EMX1 locus. And you can amplify this by PCR, and then you basically denature and then reanneal those DNA strands that are generated by PCR. And if there's an editing event that has occurred in some of those DNA molecules, when they reanneal, some of the strands will not match completely the target sequence that they reanneal with. And so you have a little mismatch in the DNA that can be cleaved by different kinds of enzymes. And in this case, we're, I think, using the T7 enzyme, but there's also a cell1, comes from celery, actually an enzyme that will cut these mismatches in PCR reannealed DNA. And so if you see, this is an agarose gel, and so if you see these smaller products running on the gel, these are evidence of an editing event that has occurred in that segment of DNA. And so you can, this isn't entirely quantitative, but you can actually get some indication of the efficiency of editing by quantifying the amount of DNA cutting that you get in these kinds of reactions. So that's just a very brief description of this type of assay. And so I'm showing you here, first of all, just two different forms of this ribonucleoprotein complex. One of them has the original guide RNA sort of construct that Martin Jinek in the lab developed, and the other is a guide RNA developed by Luke Gilbert, who's over here in the audience that you'll be hearing from later this week from Jonathan Weissman's lab, in which they had optimized the design of the guide RNA to resist to be basically expressed and more stably produced in cells. And so we compared these two, and actually in the context of this pre-assembled RNP, there's not a big difference in the behavior of these particles. You can see that you have to have a guide. If we use the protein alone, we don't see any evidence of any editing in the cells. And then this is a comparison to what we see. This is a sort of a positive control over here, delivered using a plasmid system, and then just adjusting the concentrations of the guide RNA and the Cas9, DNA encoding Cas9 in this experiment over here. So there's some variability depending on the exact ratio of guide RNA to the protein that ultimately gets produced in the cells. And one thing I want to point out is that in these experiments, we looked at cell viability as well, and we found that at least in our hands, the cells were a lot happier after receiving the pre-assembled RNA protein complex than they were after transfection with plasmid encoding these two components. And that's been pretty consistent. So, we think that that's another potential advantage of doing this, especially for cultured cells, is that, the cells are more viable after the experiment is done. This is just showing these two different guide RNA constructs that were used in the experiment. The other thing that we really wanted to do using this RNP delivery approach was to explore whether the phase of the cell cycle in which the complex was introduced to the cells might affect editing efficiency. And this kind of gets back to earlier questions about editing, about DNA repair and when that might occur and what type of repair might happen as a function of cell cycle. And so what Steve and Brett did was to use drugs that would stall the cells at different stages in the cell cycle so that they could be synchronized and then released from back into the cell cycle such that all the cells were now progressing at the same, starting at the same point in the cycle. And then we could introduce the Cas9 RNA protein complex at these different stages and ask whether there was a change in the editing or repair efficiency that occurs as a function of the timing of introduction into the cells. And so this is showing non-homologous end joining as a function of introducing Cas9 into cells that have been synchronized at the beginning of M phase. And so this is, first of all, just showing you what we see for unsynchronized cells. And these are human embryonic kidney cells that we're using in this experiment. 3 different concentrations of the Cas9 RNA protein complex that are being nucleoaffected into these cells. And you can get up to pretty decent efficiencies of non-homologous end joining, sort of about 80% in this experiment, without any synchronization. But what we found was that if we synchronize the cells first using this nicotazole, a drug that will synchronize prior to entrance into M phase, then we see that editing efficiency is much higher at lower concentrations of the RNA protein complex. We still get up to sort of the same maximum, but we get there using less of this RNP being delivered into the cells. And then finally, the other thing that we wanted to really test in this experiment was whether we could actually enhance the rate of homology-directed repair. And again, this gets back to an earlier point that came up about being able to, I think one of the real forefronts of this technology is when we get to a point where we can really control the kind of repair that happens after a cut, and furthermore, get very efficient integration of new genetic information. That will be clearly extremely powerful especially for therapeutic applications, if one could do this with high efficiency and high accuracy. And so in this experiment, the idea was to simply introduce the RNA protein complex pre-assembled into cells together with a donor DNA molecule. And we started off using just single-stranded DNAs that would introduce a restriction enzyme that would be easily detected in these reactions. And this is showing this experiment now, again, doing the synchronization of cells. So we start off with unsynchronized cells And we're including this single-stranded oligo repair template in the experiment, and cleaving with the Hindi3 restriction enzyme. So the bands we see here are just coming from molecules that actually got a Hindi3 sequence integrated at the site of the Cas9 cleavage event. And you can see that in this experiment, we get up to sort of the highest concentration of the RNA protein complex and the highest concentration of this donor template. We can get up to close to 30% homology-directed repair. But we get there, again, at much lower concentrations of these components when we first synchronize the cells. And so this was very encouraging that we might be able to actually manipulate the efficiency of this type of repair by introducing the RNA protein complex into cells at an appropriate time for that repair to be dominant in the cells. And I should say, so this was actually published at the end of 2014, and since then, it's been possible to get really much higher rates of homology-directed repair using this kind of approach, and also doing things like manipulating exactly how the single-stranded template DNA is designed, how long the homology arms are that match the sequences flanking the site of the Cas9-induced cut in the DNA. So that's what I want to say for now about delivery. And before I sort of move to the final part of this talk, I want to just ask if anybody has questions about that or comments. Yeah.
Do you know why the nucleosome is raising efficiency? Is it due to a lack of a nuclear envelope, for instance?
I think that's very possible, yeah. So to test that, we basically asked, did we get similar rates of efficiency if we just don't include the nuclear localization signal on the protein? And the answer is yes. So I think it's largely because you just get better access to the DNA. Yeah. Yeah.
Do you have any guidance with the size for the single-strand donor DNA?
Yeah. So she's asking, do we any thoughts about the size of the single-stranded DNA donor? So Steve and Brett tested DNAs ranging from 30 bases, nucleotides in length, all the way up to 200. And basically what they found was that as long as they were at least 60 nucleotides in length, so they had 30 nucleotides on either side, sort of flanking the break, they got sort of similar efficiencies of homology-directed repair. Shorter than 60 was, we saw efficiencies going down. And subsequently, I think there's some interesting data that's unpublished so far from us, and I've heard rumors of maybe this from other groups, that you can actually change the number, you can change the position of the integrated sequence and the number of nucleotides on either side of it. I'm not saying this very clearly. In other words, you could have let's say, a 60 nucleotide donor DNA where 50 nucleotides are complementary to one side of the break and only 10 or less than that on the other side, and see very different efficiencies of incorporation depending on how you skew that. Okay, so I think there's going to be some very interesting work to be done to really understand mechanistically what that means. And I don't know if Dana has ideas about that, because I don't really understand yet what that's telling us about how the repair enzymes are operating. But I think that there's going to be some very interesting data coming forward about that. I think so. Yeah, I think so. And I don't know if that's been done systematically yet, but I think that would be an obvious thing to do. It would be to really test systematically how long you need the complementarity on either side of the break in a donor template to get efficient integration.
So some of the little twists that people are seeing don't seem consistent yet.
Okay.
So I would say,
yeah.
At this point, there isn't a general way to design your donor so that it's always great. Maybe Greg Davis, when he talks tomorrow night, will have some
yeah.
things to say about that.
Right.
I did have a question about the paired
Yeah.
Has anyone explored the single-stranded oligos with the paired nucleases to see what the rules are for where it's positioned relative to the mix? I haven't seen any.
Yeah, I haven't seen that either. No, I think today the answer is no, but stay tuned. Yeah.
So, unfortunately, nicotazole is toxic in the cells I'm interested in. Are there other drugs? Because you mentioned you can get greater than 30%. What other drugs are you able to say other ways you've been able to get that?
Yeah, so have a look at that eLife paper, because we did try a series of other drugs in that paper. And if you're interested, you can also shoot me an email, and I'd be happy to put you in touch with the guys in my lab that are actually doing this. And we have some other unpublished data that we could show you that might be relevant to what you're doing. So, yeah. OK, so I'm going to move now to, I think we just have a few more minutes, right? How are we doing?
20 after.
It's 20 after. OK, so I don't want to belabor this too much, but I want to just sort of end with talking a little bit about other aspects of CRISPR biology. So Dana had asked me to give you a kind of an overview of CRISPR biology, and we talked a lot about Cas9 and where it came from and a little bit about where it's going. But I want to just kind of cycle back to some of the basic aspects of the CRISPR pathway, because I think there's still a lot of room for both basic biology to be done, as well as thinking about other applications of some of the enzymes in these pathways. And I want to share with you a short story, I'll keep it short, about how new DNA sequences are actually integrated into the CRISPR locus. This is sort of the front end of the CRISPR pathway and is really an essential part of this pathway. So, in bugs that have lost the ability to integrate new sequences into the CRISPR locus, they effectively lose the ability to use this system, right? Because if it can't acquire new sequences, then they can't adapt. And phage will very quickly develop resistance to the existing sequences in the CRISPR locus, and it becomes defunct. So, so the question is, how does that work? And so several groups, starting in around 2012, actually really earlier than that, there was some nice genetic evidence for this, but basically it was shown in early genetic experiments that two particular genes called Cas1 and Cas2 encode proteins that are somehow involved in the integration reaction. So that was shown genetically by showing that if you knock out one or the other of these genes in a CRISPR locus, those bugs lose the ability to acquire new sequences when they're exposed to a phage. Okay, so and then subsequently it was shown by several labs, including our own, that these genes encode proteins that are functional as nucleases, so they can cut DNA, they can sometimes cut RNA. It was a little bit unclear. There's a number of papers on these. It's a little bit fuzzy about what their real relevant activities might be. They're also found in all CRISPR-Cas systems, and that's actually not true for a lot of the other Cas proteins. So that was another tip-off that these were somehow fundamental to something that was central to all of these pathways. And a nice experiment done at the Weizmann by Udi Kimron in 2012, they showed that if you overexpress these two proteins in a bacterium that is otherwise doesn't have any of the other CRISPR components or Cas genes, but also has a sort of a truncated form of the CRISPR locus that looks like this, with a leader sequence and then these repeat spacer repeat units, they could actually observe integration of new DNA elements into the CRISPR locus. And so that really suggested that these two proteins might be sufficient for that reaction, or at least maybe in conjunction with other host enzymes, but at least no other Cas proteins required for that reaction. And so I'll show you some work now from our own lab to try to get into the mechanism of this. How does this actually work? And so this is the work of a student, James Nunez. And what James did was to start investigating biochemically what the function of these two proteins might be. And so this is actually showing you the CRISPR locus that's found in E. coli K12, a strain that we've probably all worked with at various times. It turns out it has amazingly, it has a CRISPR locus, but you can still transform this bug because these genes are silent. They're silenced transcriptionally. So there's a silencer that keeps them turned off so that you can these bugs can actually acquire DNA, but you can manipulate this and certainly biochemically study it And so what James Nunes in the lab showed was that these two proteins actually form a physical interaction, and he was able to crystallize that complex, and it looks like this, where he's got a dimer of the Cas2 protein in the center, and then 2 dimers of the Cas1 protein on either side. So there's 6 different proteins in this complex, 2 Cas2s and 4 copies of Cas1. And he made various mutations based on the structure that disrupt these protein-protein interfaces, and when you express those mutants in bacteria, they no longer can acquire sequences. So it looked like this really is somehow the integrase of the system. You have to have this complex forming, and the question is, how does it work? And so what James did was to set up an in vitro system where he incubated this Cas1 and Cas2 complex with DNA substrates that mimic these little DNA spacer sequences that get integrated into a CRISPR locus when the system is actually functioning. And to provide a target, he included a plasmid that encodes part of the CRISPR locus in the plasmid sequence as the target for integration, and just incubated these together. And remarkably, he actually found that there were a number of products that were produced in the presence of the Cas1 and Cas2 proteins. We didn't see anything happening to this supercoiled plasmid. By the way, this is just an agarose gel analyzing mobility of the target plasmid. And we saw really no change in the presence of either of these proteins on its own with these little DNA molecules included. But if we had everything together in the same tube, now you can see that the mobility of this supercoiled plasmid changes a lot, and we get some relaxed open circle form of the plasmid. We get something that we call band X, and we get a little bit of linearized plasmid here. And this seemed to be a function of the concentration of this substrate DNA that I'm calling protospacer DNA. So the more of that we added to the reaction, the more of these products occurred during a particular time of incubation. And I'll just summarize a number of experiments here to show you that, so first of all, if you radiolabel these DNAs, James found that these products, particularly the relaxed open circle form of the plasmid as well as the linearized form of the plasmid, become radiolabeled. And so it looked like there really was some kind of covalent interaction occurring between these little pieces of DNA and the target plasmid. And furthermore, those products were a function of the catalytic activity of the Cas1 protein, but not of Cas2. And so we could make point mutations in the active sites of each of these proteins, and only when you disrupt Cas1 do you see interference with the production of these products right here. We also had to have the intact complex of these proteins to observe these products. And importantly, the plasmid, this target plasmid, also had to be supercoiled. So if we had a pre-linearized form of this plasmid, even though the target sequence is the same, we don't see formation of these products in the reaction. And so what might these products actually be? And so we kind of imagined that for the relaxed open circle form of the plasmid, we might be getting products that would look like this, where we had one or both ends of the substrate DNAs getting ligated into the plasmid with nicking occurring as a sort of consequence of that. And for the linearized form of the plasmid, we might have something that looks like this, where we have 2 separate events. And that seemed to be consistent with the fact that that there was always a sort of a lower amount of this material, and it was a function of the concentration of the substrate DNAs that were included in the reaction. So how might this work? And so, looking in the literature, there's some nice sort of mechanistic studies that have been done on different kinds of transposases and integrases. And there are 2 kind of primary mechanisms by which these enzymes operate. One involves the formation of a protein covalent intermediate, where you have a tyrosine in the protein active site that becomes covalently linked to the integrating DNA, or sort of the target DNA during the reaction, such that you get sort of a concerted reaction that looks like this. And there's several nice examples of that kind of mechanism. And then an alternative is direct nucleophilic activation of the integrating substrate DNA, by which meaning that the enzyme actually activates the 3' hydroxyl on the end of the DNA as a nucleophile to attack a position in the target DNA sequence, leading to formation of an intermediate that might look like this, that could then be filled in and repaired in the cell by repair enzymes. And HIV integrase is one example, but there are others that work by that kind of mechanism. And so just without showing you the data, I'll tell you that James Nunes, we initially kind of favored this model because we saw several tyrosines in the vicinity of the Cas9 protein active site that looked like they might be capable of this kind of thing. But point mutations at each of those positions gave us proteins that were perfectly capable of integration. So it really didn't look like it was actually going through this kind of a mechanism. And instead, we decided to test this possibility. And this is an experiment in which James first tested single-stranded DNA substrates, which don't give rise to these products. This is what I showed you before. If we use a double-stranded DNA substrate, we get these products that I showed you. And then this is sort of a key experiment of testing a substrate DNA in which both ends have been blocked by phosphate groups that would block the 3' hydroxyls that would otherwise be on the ends of these DNAs. And when we do that, we actually see no product formation. And what was interesting was that if we did this phosphorylation on just one strand of the DNA substrate, then we see a reduced level of formation of these products. So it really looked like the 3' hydroxyls were essential in these DNA substrates, leading to the idea that this reaction might be occurring by this kind of a mechanism where we have an enzyme activating the hydroxyls on the ends of these little pieces of DNA such that they could attack a particular phosphodiester bond in the target sequence and lead to something like this, sort of these Y-shaped products. So then the question is, of course, where are those occurring in the DNA? And so James was able to generate libraries for sequencing to detect this by using either the reaction, setting up the reaction as I showed you before with this target plasmid containing the CRISPR locus, or as a control using a target plasmid that has no CRISPR locus in it. And then sequencing these products. And I'll just show you the results of those sequencing reactions. So first of all, we found that when we sequence the target plasmid that contains the CRISPR locus, very satisfyingly, a lot of these events where these little pieces of DNA are getting ligated to the plasmid occur in the CRISPR locus. And furthermore, when you look at exactly where they occur right at the junction between either this upstream leader sequence and the first repeat in the locus, or at these subsequent repeat spacer junctions. And I want you to notice too that there were sort of there were a few events outside of the CRISPR locus, but most of them, when they were outside of the CRISPR locus, occurred at a particular place near the ampicillin promoter, which was sort of a bit odd. But that turns out to be interesting because I think it gives us an important clue about how this is actually working that I'll show you in a moment. But when we look at the CRISPR sequence, this is what you see. So I didn't really emphasize this earlier, but these repeats in the CRISPR locus often, not always, but often have the ability to form, they sort of have partial palindromic character, meaning they can form these cruciforms. And we sort of imagine that if these cruciform structures can be recognized by the Cas1, Cas2 integrase complex, then that could specify the position of the nucleophilic attack at some distance away from this structure. And the reason we think this may actually be the actual target of integration is that supercoiling, in this system at least, is essential for this reaction to occur, which would be consistent with formation of these kinds of structures. If you don't have a supercoiled plasmid, then it would be very difficult potentially for these kinds of pop-out structures to actually form in the DNA. Okay, so but what about these other events that occur? And so this is actually then looking at the sequencing data for the plasmid that doesn't contain a CRISPR locus. And I don't know if you noticed the scale on the other slide, but basically, there's many, many fewer events here. So just very low levels of events in the plasmid. But again, this site near the AMP promoter was, sort of very prominent in this data. And what's interesting is that if you look at that sequence, it turns out that fortuitously it has a sequence that kind of has some of the properties of the CRISPR locus. In other words, it has an AT-rich sequence that's adjacent to a partial palindrome that could, in principle, form this kind of a cruciform structure with the integration site occurring a set distance away. And so I think it's sort of consistent with this idea that the way the system operates is by recognizing this kind of a structure. And I'm not showing you the data for this, but we have also made a number of mutations in this actual sequence, and that's also important. So it really looks like this is a system that's designed to recognize some aspects of the DNA sequence as well as its structure to specify the site of DNA integration. In our in vitro reconstituted reaction, we get products that end up like this, and we know this from various control experiments. And so I think that in the cell, then these would then be substrates for repair enzymes that would fill in and ligate and produce a fully integrated new piece of DNA. And what's really cool about this is that you can now see exactly how this works in terms of generating the templates that would be necessary to recreate the repeat on either side of the newly integrated bit of DNA by providing this single strand of the repeat on either side of the integrated product. So just going to close with a couple things here. So first of all, I think this is kind of cool because it suggests how new CRISPR loci are generated in naive organisms. So these Cas enzymes are moving around all the time on plasmids by horizontal transfer. And so you can imagine that if they get into a cell that doesn't have a pre-existing CRISPR locus or has a CRISPR sequence that doesn't work with these particular enzymes, they might, at a low level, recognize these kinds of structures in the DNA and begin inserting little bits of DNA. And of course, once you get one integration event, then it's more likely that you'll get a second one and a third one, et cetera. So you can start to see how these loci could arise in naive organisms, which I think is just, from an evolutionary perspective, is just kind of interesting and fun. And then in terms of thinking about a technology, we're very interested in the possibility that this could actually be useful for delivering foreign DNA into genomes. And there are a number of experiments going on, not only in our lab, but elsewhere, to test whether these bacterial enzymes will function in eukaryotic cells. I think the idea of being able to, so this is clearly it's a very different kind of thing than Cas9, right? Because it's not programmable the way Cas9 is. On the other hand, there might be applications where you'd like to integrate little tags into a genome to sort of map things like, sort of use it as a molecular clock, for example, or to map numbers of cell divisions and things like that, where this kind of application might actually be really useful. So, stay tuned. But I think this is something that we're sort of thinking about right now in terms of the ways that it might be useful as an application. And I really just want to close with this sort of a couple thoughts about this issue. So, thinking back to what I showed you about the germline editing of mice, and of course, you now know that there's been germline editing of humans, right? There's one paper out so far, and other labs almost certainly are working on this. And so it really begs the question, how do we think about this, and what should we do now that genomes can be edited quickly and easily? And so one of the things that the IGI has been really involved in and really interested in fostering is an open discussion about this question. And we got together a small group of scientists back in January of this year that led to publication of what we called a prudent path forward for this issue. And in particular, thinking about human germline editing, this perspective, which was published in Science magazine in March, proposed that at least right now today, scientists should agree that we're not going to use this technology for clinical applications in the human germline, just because there are important ethics that need to be discussed. We need to think about safety and efficacy, and really, are there applications where this would be the method of choice? And if there are, then how do we proceed in a safe and appropriate way? And so that has triggered it triggered a few things. It triggered a lot of headlines like this. And lots of email in my inbox and reporters calling and TV shows wanting to interview and things like that. And but fortunately, it has also spawned a very serious conversation among scientists internationally about this. And there are now in many countries, there are groups that are getting together to host meetings, many of which will be happening before the end of this calendar year. The National Academy of Sciences and the National Academy of Medicine here in the US is also involved in organizing a couple of meetings this fall that will again be international and will be focused on mostly on human germline editing, as well as maybe some other aspects of genome editing that are potentially of concern, like environmental questions that come up when you think about editing insects or plants and things like that. So stay tuned, and I certainly invite all of you to be involved in that conversation. We really want it to be open and transparent, and I think that for all of us as scientists, especially if we're using this technology, at some level we have a responsibility to be thinking about appropriate use and educating people that don't know as much about it, about how it works and how we think it should be employed in a responsible way. So that's really all I want to say. Thanks a lot to various people. I don't have the IGI specifically listed here, but certainly all of this work has been very much a partnership with my colleagues here at the IGI. This is actually my research lab here at Berkeley, and we've had great collaborators that I mentioned. I didn't mention Alan, but he's been involved in helping us with this, thinking about this integrase mechanism. And I really want to, again, just acknowledge the IGI, and we've had the great good fortune to have Dana Carroll here on sabbatical for a few months, and he was the one that really organized this whole workshop. So big thanks to Dana. And of course, if you have other questions or want to discuss further, I'm here. Thanks a lot.